Hello friends, welcome back and let's start our next session on DNA. We'll be starting this time from the DNA. Okay, where is the live stream? Where is the live video? Okay, this is live. Here we go. Okay, so like I said, we will start with the DNA. Before I go there, remember vitamin K is required for activation. It is required for activation of clotting factors. This means deficiency will cause hemolysis. Rather, you can say hemolytic tendency. Commonly, we call it the bleeding diathesis. Not hemolysis exactly, but bleeding diathesis means when there is injury, the tendency to clot will be decreased, but it's not hemolytic per se. Means hemolytic anemia. Okay, this I'll check for you about hemolytic anemia. But as I told you, vitamin E is not associated very strongly with any specific disorder. Vitamin E deficiency only has, you can say, general presentations. It doesn't have any specific findings. Vague presentations are there. More so because vitamin E deficiency is very, very rare. Any foodstuff that you're consuming will have significant amount of vitamin E. So vitamin E not associated with any specific well-defined functions except for the antioxidant activity, and therefore not associated with the disorders. Only thing we can think about is the antioxidant function. The antioxidant function, because it is not there, may result in hemolysis. I'll check it and let you know. Only thing which is possible is the antioxidant function may cause the hemolysis, but I'm not sure. I'll check it out. In that case, we will be talking about vitamin E. Okay, so let's go back to our session. We're talking about the DNA. Talking about DNA. Now, when we talk about DNA, we'll be talking about the different types of DNA. That is the nuclear DNA, the nuclear DNA, the mitochondrial DNA. Anywhere else? Do we have DNA anywhere else besides the nuclear and the mitochondrial DNA? Do we have any other DNA? Yes, we have what is known as the cell-free DNA. Please understand. Please understand. Just like after the damage to the cells, the cellular content comes out in the form of proteins. The different enzymes are there whenever there is a cell turnover. In the same manner, the DNA can also come out, and this is what is known as the cell-free DNA. The cell-free DNA has become very important for your exam because you should know uh, two new modalities which make use of cell-free DNA. Two new modalities which make use of cell-free DNA. First is the liquid biopsy. First is the liquid biopsy. Basically, the liquid biopsy is used to identify malignancies using blood sample. Obviously, this is done after imaging studies when we find some mass in a place which is not easily accessible. For example, in the retrosternal space, we cannot easily go into the retrosternal space. At the back, there is spine. At the front, there are the large vessels and the heart. So it is not easy to reach in the retrosternal space. So in that cases, when we have some mass and we have a probable list of differentials, then rather than take the sample, what we do, take this sample, blood sample, and we separate out the cell-free DNA and we look for malignant transformation. We look for malignant transformation in the cell-free DNA. Right. In this way, we are able to identify the malignancy. The second very important use of the cell-free DNA, second very important use of the cell-free DNA is what is known as NPT or NIPT, which stands for the non-invasive prenatal testing. Stands for the non-invasive prenatal testing. Okay, so what is non-invasive prenatal testing? This is a new test which has been launched just 4 or 5 years back. It became available in India in 2016 only. The non-invasive prenatal testing is used to identify trisomies. So how do we do it here? In the mother's blood, if you take up the cell-free DNA, majority of it is maternal in origin. Majority of it is maternal in origin, but about 10% is fetal in origin. About 10% is fetal in origin. Alright, and we separate this 10% fetal DNA, and then we do gene sequencing for the target chromosomes. We identify certain specific genes. We identify certain specific genes for the three chromosomes, the chromosome number 13, 18, and 21, and we do the gene sequencing. The normal amount is considered as 100%, whereas in trisomy the amount of that particular gene will become 150%. So this is how we can identify this is how we can identify the trisomies. Please note that the NIPT is a NIPT is a screening test with high sensitivity. Sensitivity is greater than 99%, but confirmation by either chorionic villus sampling or amniocentesis, depending on stage of pregnancy. Confirmation can only be done by CVS or amniocentesis. However, the screening can be done by the non-invasive prenatal testing. So these are two new important tests which you have to keep in mind. The NIPT uses the cell-free DNA of the fetus in the mother's blood. It uses the technique of gene sequencing. It uses the technique of gene sequencing. In a positive sample, we will have approximately 150% of a given gene from one of the three chromosomes. Sensitivity is very high, greater than 99%, but confirmation must be done, which is done either by the chorionic villus sampling or the amniocentesis. The confirmation is done either by chorionic villus sampling or by the amniocentesis. So this is about the cell-free DNA. Now let us first talk about the nuclear DNA, and then we'll talk about the mitochondrial DNA. So let's move on to nuclear DNA. Okay. So when you talk about the nuclear DNA in the humans, it has a large number of different conformations, but the most common conformation is the B form. The most common conformation is the B form of DNA. Besides the B form, the different forms which may be seen are the A form, the C, D, and E form. But the second most common form after the B form is the Z form. So the two common forms are the B form and the Z form of DNA. The two common forms are the B form and the Z form DNA. The others A, C, D, and E are quite rare. The A, C, D, and E are quite rare. Okay, so let us first talk quickly about the B form DNA, and then we come back. Then we talk a little bit about the Z form DNA. Okay, the B form of DNA, the most common conformation, the most common conformation, it is double stranded. The two strands are antiparallel. The two strands are antiparallel. The nucleic acid is linear. Why I'm saying linear? Because when you go to the mitochondrial DNA, it is circular in nature. Okay, so linear. Each of the strand is present as a helix. So we have what is known as the double helical structure. It is a double helical structure. But whenever you talk about helices, you have to tell us whether the helix is right-handed or the left-handed. So this is a right-handed helix. Lastly, it shows the presence of grooves. The presence of grooves, we have the major and minor grooves. We have what are known as major and the minor grooves. So the B form, double stranded antiparallel linear double helical structure, right handed, showing grooves, the major and the minor grooves. If you look at the structure, the structure is something like this. The structure is something like this. The two strands of DNA running together in the helical form. Now, in this structure, a few things of note which you should be able to tell us. Number one, number one, what is the width of the helix? What is the width of the helix? So when we ask about the width of the helix, the answer is, depending on the unit, 20 Armstrong or 2 nanometer. The width of the helix is 20 Armstrong or 2 nanometer. The second part is, what is the distance occupied by one turn? What is the distance occupied by one turn? Now here there is a lot of confusion. Different books might give different answers. So I'll just quickly tell you. Most of your UG level books will give you the answer as 34 Armstrong having 10 base pair per turn. 34 Armstrong having 10 base pair per turn. But please note, the more correct answer would be 36 Armstrong having 10.5 base pair per turn. The more correct answer will be 36 Armstrong having 10.5 base pair per turn. Uh, for UG level, we mentioned 34 Armstrong. So if 36 Armstrong is not there in the option, you obviously have to go with the 34 Armstrong. Or if both are given, then 36 Armstrong is definitely the better answer. If both options are there, then 36 Armstrong is the better answer. If 36 Armstrong is not there in the question, then you go with the 34 Armstrong. And this is known as the pitch of the DNA. The distance occupied by one turn is known as the pitch of the DNA. Alright, so for the B form of DNA, the double stranded arrangement, the two strands are anti-parallel, thus DNA is linear in nature. The structure is the double helical structure, which is a right-handed helix, and there are major and minor grooves. When I say right-handed, it means the winding of the helix is clockwise. The winding of the helix is clockwise. We quickly compare this with the Z form of DNA. The Z form DNA, it is the second most common. It is the second most common. The first three are the same. Double stranded, anti-parallel, linear. Basically, they are the properties of DNA. They are the same. What will change will be the conformation. It doesn't have a double helical structure. It has a zigzag arrangement. It has a zigzag shape. It is left-handed, and there are no well-defined grooves. There are no well-defined grooves. In other words, the first three are the same. The next three are different. The next three are different. If you look at the structure, why we call it the zigzag, and that is why we call it the zigzag structure. and from where we get the name zDNA. The pitch of zDNA is 12 base pair per turn. If we talk about the pitch, it is 12 base pair per turn. The pitch of the Z form is 12 base pair per turn. So Z form, double stranded, the two strands are anti-parallel and linear. It has a zigzag shape. The DNA is left-handed and there are no grooves. The pitch of the DNA is 12 base pair per turn. Talking about the A, C, D and E forms, you can note the A form will have 11 base pair per turn, whereas the C, D and E forms, each of them will have 8 base pair per turn. Each of them is going to have 8 base pair per turn. So this is a little bit about different conformations of the nuclear DNA, the B form, the Z form, A, C, D and the E forms. Now besides this, the second part that we have to understand is the arrangement of nuclear DNA. After this we have to know the arrangement of nuclear DNA. Again, this is very important because if you compare the shape of the cell with the length of the DNA, then there is a great mismatch. Size of the cell in which the DNA is there is typically in the range of 5 to 60 microns, whereas the total length of DNA, the total length of the DNA is in the range of 2 meters. So you can just compare, the size of the cell is 5 to 60 microns, 10 to the power minus 6 meter, and the total length of the DNA is 2 meter. And this cell that I'm talking about, this is the complete cell. In this complete cell we have a small part which is the nucleus, and within the nucleus also all the DNA is concentrated in a small part. You can imagine the DNA must be compacted to a very great degree before it can successfully be stored inside the cell, and that is what we call the arrangement of nuclear DNA. The DNA is arranged at multiple levels. Typically we will find 4 to 6 levels. In the first level, the arrangement is of a solenoid. The solenoid is then arranged as a super solenoid. The solenoid is then arranged as a super solenoid. The super solenoid gives way to loops. The loops give way to whorls. The whorls are again arranged as solenoid, and finally the super solenoid. These are the shapes that we'll get to see at the different levels. After every level there is compaction of the DNA. After every level there is compaction of the DNA. Luckily for us, we need to focus only on the first and the last level. We can skip out the levels in between. We only need to focus on the first and the last level. The first level, the shape is known as the nucleosome. The last level, the shape is known as chromatin. The chromatin is further compacted and arranged as chromosome but only in the M phase. Only in the M phase we have the chromosome. Only in the M phase we have the chromosome, but in all other phases we will have the chromatin, which is the final shape. So what we have to do, we have to talk about the nucleosome and the chromatin. The other intermediate steps can be ignored, they are not frequently asked. Okay, so let's first talk about the nucleosome. Okay, so we are talking about the nucleosome, and as mentioned, the nucleosome is going to have the solenoidal arrangement. What is a solenoid? Anyone has idea what is a solenoid? Basically, a cylinder around which the wire is wrapped. This is a solenoid. We had seen this in physics, in electricity, when we were in 11th and 12th. Solenoid, a cylinder around which the wire is wrapped. Coming to the DNA, here also the similar structure is there. We have a central cylinder like structure. We have a central cylinder like structure around which the DNA is wrapped. Around which the DNA is wrapped. Alright. This cylinder, this cylinder is known as histone octamer. As the name should make it clear, there are 8 subunits in here, and that is why we call it the histone octamer. The 8 subunits are H2A, H2B. They are paired up as a dimer. H2A, H2B, H3 and H4. H3 and H4, which is paired. Everything is double. 2H3, 2H4, 2 copies of the dimer make up the histone octamer. 2 copies of H2A, H2B, H3 and H4 make up the histone octamer. 2 copies of H2A, H2B, H3 and H4 make up the histone octamer. We'll talk about histone octamer in a short while. Let's complete the structure and then I'll talk about the histones that we have seen here. The DNA which is attached here, the DNA which is attached here typically forms 1.6 base pairs. Now obviously this nucleosome cannot be sitting alone. It cannot be sitting alone. So there will be other nucleosomes nearby. There will be other nucleosomes nearby. And they have a similar structure. A similar structure will be seen in all the nucleosomes. And because they are part of the same DNA, they are in continuation. They are in continuation. The segment of DNA which is in between the two nucleosomes is also attached to another histone. It is also attached to another histone, which is the H1 histone. What is the H1 histone? The intervening DNA between the two nucleosomes is known as the linker DNA. It is known as the linker DNA. Linked DNA is commonly described to be having 54 base pairs, but in some books the value is mentioned even as 30 base pairs. So depending on the options, preferred answer is 54 base pairs, but in some books it will say the 30 base pairs. All right. So this is the typical structure of nucleosome as seen under as seen under contrast electron microscope, as seen under the contrast electron microscope. This is a detailed version of the electron microscope where the computer fills in the colors on its own by depending on the contrast. So we have the contrast type electron microscope with the detailed nucleosomal structure where we can see the histone octamer and we can see the DNA looping around the histone octamer. We can see the linker DNA. If we don't have the contrast type of electron microscope, but rather you have the regular electron microscope. If you just have the regular electron microscope, what you are going to get is something like this: nucleosome, linker DNA, 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 something like this. This is described as beads on string appearance. So under the regular electron microscope, we get to see the beads on string appearance of the arrangement of DNA, whereas under the contrast electron microscope, we get to see the nucleosomal features. All right. Now, when there is cell damage, when there is cell damage, the cleavage of the DNA also follows the pattern of cell damage. You know, there are two types of cell damage. You can have the apoptosis and you can have the necrosis. In case of necrosis, a pattern or a uniform mechanism to break the DNA is not maintained, whereas it is seen in case of the apoptosis. So like I said, you can have apoptosis. In case of apoptosis, the cleavage occurs in between the nucleosomes. The cleavage occurs in between the nucleosomes. It doesn't disrupt the nucleosomal structure. So depending on the distance between the first and second cleavage, imagine the first cleavage is here and the second cleavage is here, you'll get 3 nucleosomes. If the second cleavage is here, you get 2 nucleosomes. If the second cleavage is here, you get only 1 nucleosome. Because of this, in a process, when you perform the gel electrophoresis, when you perform the gel electrophoresis, the pattern for DNA that you get, the pattern for DNA and that you get, if the well is here, if the well is here, the pattern that you get is something like this. We call it the ladder pattern. But in necrosis, the cleavage can occur anywhere. The cleavage can occur within the nucleosome also. There is no sanctity of the shape of there is no sanctity of the shape of the DNA, and that is why if you have the well here, in this case what you get is a pattern like this. The nucleic acid of all sizes may be seen because the cleavage can occur anywhere, and This is what we call as the smear pattern. So in our process, the ladder pattern that you see is actually multiples of nucleosome. The multiples of nucleosome meaning the 54 base pair and the 146 base pair, approximately 200 base pair per nucleosome. You are going to get the 200 base pair. Multiples of 200 base pair are seen in the ladder pattern, and then we have the smear pattern. Please note that these ladder and smear pattern are not exclusive for apoptosis and necrosis. Sometimes in apoptosis you may see the smear pattern, and sometimes in necrosis also you may see the ladder pattern, but more or less this is the trend that we see in apoptosis and in the necrosis. This is about the nucleosomal structure. Now let us talk a little bit about the histone. Okay, so we're going to briefly talk about the histones. Remember, the DNA will have large number of phosphates, and therefore the DNA will generally be negatively charged. Because of the large number of phosphate, the DNA will be negatively charged, and to attach to that DNA, the protein which are attaching must be positively charged. In other words, they are basic in nature. They are basic in nature. The basic nature is due to presence of the amino acid lysine and arginine. Please note that histidine is not present in any great amount. Although the names sound similar, histidine, histone, it is not so. The basic nature is imparted by lysine and arginine. They are responsible for the basic nature. We already mentioned that there are 5 types of histone. Okay, I didn't label the histone here. Okay, I labeled it. Yeah, there are 5 types of histone: H1, H2A, H2B, H3, and H4. H1, H2A, H2B, H3, and H4. These histones, even when they are attached to DNA, can undergo covalent modification. This covalent modification is important because it defines their interaction with the nucleic acid. So what are the different types of covalent modification that they can undergo? The most well studied is the acetylation. The most well studied is the acetylation. In addition to that, we can have the phosphorylation. We can have ADP ribosylation. We can have the ADP ribosylation. We can have methylation. We can have methylation. We can have monoubiquitination. The attachment of single ubiquitin is known as monoubiquitination. Then another change is known as sumoylation. Addition of sumo protein is called sumoylation, and lastly we have what is known as citrullination. SUMO stands for small ubiquitin related modifiers. Small ubiquitin related modifiers is SUMO. Attachment of SUMO is known as SUMOylation. Addition of SUMO is known as SUMOylation. So all of these are the covalent modification which can occur in the histone. The net result of all these modification is they modify the basic nature. They modify the basic nature of the histone. They can increase as well as decrease the basic nature. Now this is very important. See, the DNA is wrapped around the histone and it is tightly attached because they have the opposite nature, so they are tightly attached. Now if I make the histone less basic so that the attraction is less, the separation of the DNA from the histone will become easier. If the separation is easier, the transcription will occur faster. So by modifying the basic nature, we can regulate the rate of two things, primarily transcription, but to some extent replication also. Primarily transcription, but to some extent we can regulate the replication also. Just like we have the covalent modification of the histones, we also have the DNA methylation. Please note, in the DNA there is only one possible change, and that is the DNA methylation. In the DNA we have only one possible change, that is DNA methylation, and it also contributes to regulating the rate of transcription and replication. This regulation of transcription replication by covalent modification is what is known as epigenetics. The regulation of transcription and replication by covalent modification of histone and DNA is known as epigenetics. So essentially what is happening here, we are altering the interaction between the histone and the DNA, and that is how we are altering the rate of transcription and replication. Please remember that epigenetics is inheritable. The other term used for inheritable is transmissible. The epigenetics is inheritable, or in other words it is transmissible. Okay, so what is the importance of epigenetics? Why I'm mentioning about this? Epigenetics depends on environmental factors. What is happening here, we are not changing the gene sequence, whatever change is occurring is the covalent modification of the protein and the DNA, which is outside the gene sequence. Here the gene sequence will still remain the same, but still there is some change in the transcription and the replication. That is why we call it the information beyond the genetic information. The regular information in the sequence of nucleotide is the genetic information, and this is the extra information which is present by covalent modification, and it depends on environmental factors. In other words, the epigenetics is the gene environment bridge. It is the gene environment bridge. What this means is, previously the concept was that whatever phenotype that we see is directly coming from the sequence of nucleotides, which we call the genetics. But now it is understood that okay, the sequence of nucleotides is very important to give us the phenotype, but what is getting expressed to what intensity, with what frequency, that is not just a factor of the genetic sequence. It can be affected by the covalent modification of the nucleotides and the histones, which is known as epigenetics. This means that phenotype, as we see, is a sum of genetics, what all genes are there, and epigenetics, what all genes are getting expressed and at what frequency. So the final phenotype that we see depends on both the factors, the genetics and the epigenetics. So this is the part that you need to remember. The phenotype of a person depends on the genetics as well as epigenetics. Epigenetics itself depends on the environment. So this represents the gene environment interaction or the gene environment bridge. That is what is epigenetics. Nowadays we have the term called epigenome, and the study of this epigenome is known as epigenomics, because now a lot of association has been established between epigenetic changes and the causation of disease, particularly the chronic disorders. So a lot of evidence has come in the association between epigenetics and various disorders. So the whole set of epigenetic changes is known as the epigenome, and the study of the epigenetic changes is known as epigenomics. So you should remember the terms epigenetics, the epigenome, the epigenomics. The phenotype being a result of gene environment bridge, that is the sum together of genetics and the epigenetics. The epigenetics itself is, although it is depends on the environmental factors, is inheritable as well as transmissible. The epigenetics is inheritable as well as transmissible. So this is about the histones. As you should know, the histones are positively charged or basic in nature proteins. These are proteins which are positively charged or basic in nature. The basic nature is due to the presence of lysine and arginine. The five types of histone are there: H1, H2A, H2B, H3, and H4. The histones show covalent modification. The different types of modification include the acetylation, the phosphorylation, the ADP ribosylation, the methylation, the mononucleotylation, sumylation, and the citrullination. All of these can modify the basic nature, either increase or decrease depending on type and where it is occurring. By this modification of basic nature, we regulate the rate of transcription and replication in addition to the DNA methylation. Together, this is known as epigenetics, which is inheritable and transmissible. And the epigenetics depends on environmental factor, therefore, it represents the gene environment bridge. The epigenetics tells us that the final phenotype will be a sum together of genetics and the epigenetics. The complete epigenetics of a individual organism is known as epigenome, and the study of epigenetics is known as epigenomics. So this was the first level of organization, which we called the nucleosome. Let's go to the last level of organization, which was the last level of organization, which was the chromatin. So now we are going to be talking about the chromatin. The chromatin can be of two broad types. The two broad types, the euchromatin and the heterochromatin. The euchromatin and the heterochromatin. The heterochromatin itself is of two types. We have the facultative and the constitutive. So euchromatin, heterochromatin can be facultative or constitutive. The constitutive heterochromatin may be microsatellites, or they may be satellites. The satellites include the telomere and the centromere. So this is the description of the chromatin. We have the euchromatin, the heterochromatin. Heterochromatin can be facultative or constitutive. The constitutive chromatin may be microsatellites or satellites. The satellites include the telomeres and the centromeres. They include the telomeres and the centromeres. Let's talk about them one by one. Okay, so this is a segment of DNA which is also undergoing transcription at one place. It is undergoing a transcription at one place. Alright, so the place where transcription is going on, this is the segment of DNA which we call as the Euchromatin. In other words, it is the true chromatin or the true DNA. Wherever gene will be there, that is the segment we call as the true DNA. And where the transcription is not going on, we call it the heterochromatin. Now remember, the DNA absorbs light at 260 nanometer, therefore obviously it will not be visible to the naked eye. To visualize this DNA, what do? We label it with a dye. Let's say we are going to label it with the dye like this. The dye will get attached to the DNA at equidistant positions. It will get attached to the DNA at equidistant positions, not only on one strand but on both the strands. On equidistant position, it will get attached to the DNA. Right? Now, when you examine under the microscope, when you examine under the microscope, when you examine under the microscope, let's say you get the segment like this, and I'll shift it to a little bit to the left. Let's say you get a segment like this, and another segment that you get is like this. You can see similar in shape. Now the DNA will not be visible. What will be visible will be only the dye. Okay, so what you find, the euchromatin appears to be lightly staining. The dye particles are very far apart. It is lightly staining, whereas the heterochromatin is darkly staining. The heterochromatin is darkly staining. The dye particles are close together and it gives the appearance of darkly staining. This is because in the euchromatin we saw the transcription was going on. It is transcriptionally active. It is transcriptionally active, whereas the heterochromatin is transcriptionally inactive. The heterochromatin is transcriptionally inactive, and because it is transcriptionally active, the euchromatin is uncondensed, whereas the heterochromatin is condensed. So these are the three important features to distinguish between the euchromatin and the heterochromatin. The three important features to distinguish between euchromatin and the heterochromatin. Talking about the heterochromatin, the facultative and the constitutive heterochromatin, the example of facultative heterochromatin are the bar bodies. The example of facultative heterochromatin are the bar bodies. The microsatellites are typically nucleotides having 2 to 6 nucleotide length which are repeating again and again. They are repeats having 2 to 6 nucleotides. The telomere and centromere are bigger repeats. The size of the repeats in the telomere and centromere is much bigger than 2 to 6 nucleotides. Okay, one example of microsatellite repeats could be the triple nucleotide repeats. The triple nucleotide repeats can be an example of the microsatellites. So I'll repeat. The chromatin can be euchromatin or heterochromatin. The heterochromatin may be a facultative or constitutive. Example of facultative heterochromatin is the bar body. The constitutive heterochromatin may be microsatellites or satellites. Example of microsatellite are the triple nucleotide repeats. Normally they can vary from 2 to 6 nucleotide in length, which are repeating n number of types. The satellites include the telomere and the centromere. Little bit about the telomere and the centromere. Little bit about the telomere and centromere. Okay, there was a temporary net failure. I think it should come back very shortly. So you haven't missed anything because I was watching the feed and I have not mm, uh, given any content when the net went out. Don't worry, you have not missed anything. I have not given any extra content. So like I said, now let us talk a little bit about the telomere. I'll talk a little bit about the telomere. You have not missed anything. We did not relay anything when the uh, buffering was going on. Okay, telomeres, it is the end of chromosome. It is the end of chromosome. It has a very specific sequence. It has a repeating self complementary sequence. It has a repeating self complementary sequence. We'll see what is the importance of this repeating self complementary sequence and what are its functions. It stabilizes the ends of DNA. It stabilizes ends of DNA and it protects from DNA repair machinery. It protects from DNA repair machinery. So telomeres are end of chromosome. They are repeating self-complementary sequence, and they protect from DNA repair machinery. They stabilize the end of DNA. What do you mean by they protect from the DNA repair machinery? See, this is end of one DNA. This is the end of one DNA. You will note that one of the strands is longer than the second strand. It is a property of the end of DNA. Why it happens? We'll see this when we are doing the replication. Don't worry about that. We'll see why it happens when we are doing the replication. Another end of DNA. The same thing. The same thing. We can see one end of DNA is longer than the second end. One end of DNA is longer than the second end. Now, uh, when the DNA repair machinery comes across the two DNA like this, DNA repair machinery. See, I have drawn the two DNA strands with separate color, but in the cell the DNA will be of same color. There is no different color for the different chromosomes, so they are of the same color. And when the DNA repair machinery looks at the two DNA which are sitting together and the ends are sitting together, what it thinks? Oh, this DNA has got broken. I must repair this DNA, and it will try to bridge the gap. It will try to bridge the gap, and thereby it will try to join the two strands of DNA. The DNA repair machine will try to join the ends. Alright, but what is the problem here? When you join the ends of DNA, we have the guardian of genome P53. When you are in the hostel, there is a random roll call. There's a roll call at, let's say, 8:45. How many are there in the hostel? How many have bunked away? And immediately when the 53 does the count, instead of 46 chromosome which should have been there, we will find only the 45 chromosome, and it will sound the alarm that irreversible damage has occurred. One of the students has run away from the hostel. So because the count of the DNA is changed, which cannot be corrected by the 53, it will induce the apoptosis. So we have to protect the ends of DNA from DNA repair machinery. We have to protect the DNA from the DNA repair machinery. How it happens? By the organization of the DNA. See what happens. This is one of the ends of DNA. One of the ends of DNA. Now this DNA folds upon itself. This DNA folds upon itself. When it folds upon itself, the change is something like this. Just in the preceding part, because of the pressure of folding, there will be slight unwinding, and the smaller end comes to rest upon itself. The longer end will also fold upon itself, and it comes to lie in the part where the denaturation has occurred. Now recall that the sequence of the telomere is self complementary. This means the segment which has come inside is complementary with the preceding sequence of the DNA, and therefore it is able to form the hydrogen bond. It is able to form the hydrogen bond, and now when the two ends of DNA will come together, now when the two ends of DNA will come together, the DNA repair machinery cannot join the two ends. This is how the telomere protects from the DNA repair machinery. One more point to note is the pairing that we see here is the Chargaff base pairing. In addition to this Chargaff base pairing, there is a non-specific pairing at the other end also. There is a non-specific pairing at the other end also. This is described as the Hogstein base pairing. So where do you find the Hoxton base pairing in the DNA? The answer is the telomere. The Hoxton base pairing is found in the DNA in the telomere. Because of the Hoxton base pairing, if you look at this particular segment of DNA, if you look at this particular segment of DNA, you will find triple stranded DNA. Where do you find the triple stranded DNA in the cell? You find it in the telomere. The preceding segment resembles the letter D, and therefore it is commonly described as the D loop. Where is the D loop seen in the DNA? The answer is the telomere. So I'll repeat. Hoxton base pairing, where do you see it? We see it in the telomere. Triple stranded DNA, where do you find triple stranded DNA? We find it in the telomere. Where is the D loop in the DNA? The D loop is present in the telomere segment. So these are some of the important points about the telomere. Some of the important points about telomere. Remember the functions of telomere. It stabilizes the ends of DNA and it protects the DNA from DNA repair machinery. If we are unable to do this protection, the two DNA will combine together and will result in apoptosis. This is what happens when the telomere is deleted. We'll see later when the replication occurs, the telomere is deleted. Once all the telomere is deleted, there will be tendency for the two ends of DNA to join under the influence of DNA repair machinery, and the cell will undergo apoptosis. That is why once the telomeres are completely exhausted, the cell undergoes apoptosis. Okay, so this is telomere. Make extra note of the sequence. It is a repeating self complementary sequence which allows for the binding of the folded segment. On the same pattern, let us quickly mention a few points about the centromere. A few points about the centromere. Centromere is the so-called center of DNA. It is the so-called center of DNA. Label it as a so-called center of DNA because it is not always in the center. It may be in the center. It may be slightly shifted from the center, submetacentric. It may be tilted towards the end, acrocentric, or it may be right at the tip of the end when we call it the telocentric. So the centromere need not always be at the center. It can be metacentric, submetacentric, acrocentric, or telocentric. What is the primary function of the telomere? Provides what is known as docking site. For whom? For whom? Provides docking site for microtubules. When the cell division is going on, mitosis or meiosis, we have the chromatids. The two chromatids are joined at the center, and they have to be separated. They have to be pulled and taken to the poles of the cell, which is done by the microtubule. So they have to attach somewhere. We call that point the docking site, or in simple words, it is the point of attachment. So the centromere provide point of attachment for microtubules in cell division, which allows for the two chromatids to reach the pole. One very important point: the telomere and centromere are structural units. They do not have any relationship with the gene, either regulation or the transcription, and therefore they are not related to inherited genetic disorders. They are not related to inherited genetic disorders means if there is some mutation in this area, it is not going to cause some type of genetic disorder. These areas do not contain gene. Remember, they are the constitutive heterochromatin. There is no gene here, there is no transcription going on. In addition, they do not provide any type of regulation also. In contrast, in contrast, the microsatellites, they are regulatory elements. They bind to regulatory proteins, and because of this, they are frequently associated with inherited disorders. They are frequently related with inherited disorders. I had given you the example that they may be the triple nucleotide repeat, and therefore they are frequently associated with the triple nucleotide repeat disorders. You will discuss a list of these disorders when we are doing the genetics in the pathology. When we are doing genetics in pathology, they will talk about a large number of triple nucleotide repeat disorders. Huntington's chorea, the Fragile X syndrome, all of these are examples of the triple nucleotide repeat disorders. Okay, so this is about the nuclear DNA. This is about the nuclear DNA. We said the nuclear DNA has multiple levels of organization. First, we talked about the nucleosomal level. Then we talked about the chromatin. In the chromatin, we talked about the euchromatin, heterochromatin. In the heterochromatin, we talked about the facultative and the constitutive. In constitutive, we had several names: the microsatellite, telomere, and centromere. I briefly mentioned the difference between euchromatin and heterochromatin. Then I talked about the telomeres. What are telomeres? And then I mentioned the details of the centromere. The telomere and centromere are not related or associated with inherited genetic disorders, whereas microsatellites, because of their regulatory function, may be associated with inherited disorders. The classical example being the triple nucleotide repeat disorders. So this covers the nuclear DNA. Let us quickly cover the mitochondrial DNA. Any question you want to ask so far from the nuclear DNA before I start talking about the mitochondrial DNA? Any question you want to ask from the nuclear DNA? Any questions from the nuclear DNA before we go on to the mitochondrial DNA? If there are no questions, then I'll quickly move on to the mitochondrial DNA. If there are no questions, I'll quickly move on to the mitochondrial DNA. The mitochondrial DNA, like the nuclear DNA, is double stranded. But unlike the nuclear DNA, it is circular. In addition, it is quite small, having 16569 base pairs. H1 is not there in histone octamer. We had the H2A, H2B, H3 and H4. H1 is not a part of the histone octamer. H1 is not a part of histone octamer. It is a part of the separate linker DNA. It is associated with the linker DNA. In histone octamer, we have H2A, H2B, H3 and H4, 2 copies of each. The 16569 base pairs code for "37 genes". Together they code for 37 genes. 13 are for proteins. 2 are for ribosomal RNA, and the remaining 22 are for the transfer RNA. Remaining 22 are for the transfer RNA. Please note, all the 13 proteins belong to the electron transport chain. The electron transport chain itself has 67 protein subunits, out of which only 13 are coming from the mitochondrial DNA. Only 13 are coming from the mitochondrial DNA. In other words, the contribution of the mitochondrial DNA for proteins of ETC is less than 20%. For other systems it is 0. For other systems it is 0. For ETC it is less than 20%. For ETC it is less than 20%. So small size of the DNA, it is 16569 base pairs, having a very limited number of genes. However, we don't have introns here. We don't have introns here. No introns are there. A very important implication of this information is, because introns are not there and the nucleic acid is not present in a compartment, is not present in the nucleus, it is present in the cytoplasm, where the transcription will occur and the translation will also occur. As a result of this, simultaneous transcription and translation can occur. So the transcript that you get is ready for translation. The transcript that you get is already ready for translation. You don't need to modify it. The introns are not there, so there is no cleavage required, joining of exons, etc. That part need not be done. In addition to no introns, we have very few untranslated sequences. We have very few untranslated sequences. In comparison, in nuclear DNA, only 1.5% of DNA is translated. So you can compare, in the mitochondrial DNA almost all of the DNA is translated, whereas in the nuclear DNA only 1.5% of the DNA gets translated. The mutation rate in comparison to nuclear DNA is higher, about 5 to 10 times of nuclear DNA. The mutation rate is 5 to 10 times in comparison to the nuclear DNA. There is a very high mutation rate. Lastly, there is some variation from the universal genetic code, so we call it the exceptions to genetic code. What are the exceptions that we see to the genetic code here? To list this exceptions to genetic code, what I'll do, I'll write the codon that we are talking about. We'll mention the normal translation, and then we'll mention what we see in the mitochondria, right? Okay, so we start with the UAG. UAG is normally a stop codon. You know that UAG is normally a stop codon, but in the mitochondria it codes for Tryptophan. Not UAG, sorry, it is UGA. UGA is normally a stop codon. In mitochondria, it codes for tryptophan. At the same time, AGA and AGG, AGA and AGG, which normally code for arginine, in the mitochondria they work as stop codon. In mitochondria they work as stop codon. Then AUG, which normally codes for methionine, codes for formyl methionine in the mitochondria. It codes for formyl methionine in the mitochondria. And lastly, AUA, which is normally isoleucine, also codes for methionine in the mitochondria. So these are the exceptions to genetic code that we see in the mitochondria. Normally, the translation of each of the codon in any biological system should be the same, but there are certain differences that we can see in the mitochondrial translation. So these are the exceptional genetic code that we see in the mitochondrial DNA. Lastly, the inheritance pattern of mitochondrial DNA is unique, which can be very easily understood when we talk about the mitochondrial DNA disorders. And the disorders is why the mitochondrial DNA has become important. See, previously it was assumed that all the genetic disorders are related to the nuclear DNA, because the mitochondrial DNA contribution was very small. It is less than 0.1% of the total DNA which is present in the cell, and moreover it was supposed to produce a very little number of the proteins, so it was not considered to be important. But in the last one and a half decade, a large number of disorders have been shown to be associated with the mitochondrial DNA, defect in the mitochondrial DNA, and now these are identified as mitochondrial DNA disorders. These are now identified as the mitochondrial DNA disorders. Okay, so when we talk about mitochondrial DNA disorders, the first thing that we should understand is the inheritance pattern of disorders. Inheritance pattern of these disorders. The inheritance pattern depends on two very important points which you have to remember. See, from where does the mitochondrial DNA come in the newborn? What is the source of mitochondrial DNA? Does it come from the mother? Does it come from the father? Or it comes from both? My question is, source of mitochondrial DNA. Maternal, paternal, both, either. It is not fixed, it can come from any of them. It is not fixed, it can come from any of them. What is the source of the mitochondrial DNA? Anyone? So when you talk about the source of mitochondrial DNA, it is very important to understand, like you said, that all mitochondrial DNA will come from the mother only. all mitochondrial DNA will come from mother only. What happens when the sperm and ova fuse? From the sperm only head goes. The body and the tail is left behind, which contains all the mitochondria. So only the nucleus is transmitted to the ova for formation of the zygote, and all the mitochondrial DNA is left behind. Only the maternal origin of mitochondrial DNA is there. Okay, so the second question is, suppose we have a cell. Suppose we have a cell which requires more mitochondria. Now you have started having more activity. Suppose you start having some uh, uh, fancy of doing exercise. Now there's a lockdown, so you want to become very very fit. You have allowed yourself to become unfit when the normal days were there. So during the lockdown you are doing a lot of exercise. When you're doing a lot of exercise, the cells will require more and more of mitochondria. So how to generate or obtain new mitochondria? Suppose a cell requires more mitochondria, your muscles will now require more mitochondria. So how are you going to generate those new mitochondria or obtain those new mitochondria? One is borrow from neighboring cells. For example, adipocytes are there, they don't require the mitochondria, so you can borrow them from the adipocytes. You can synthesize using existing mitochondria as template, or you can have what is known as binary fission. Lastly, any of the above methods may be used to obtain the new mitochondria. Any of the above may be used to obtain the new mitochondria. What is the answer? What is the answer? How to generate or obtain the new mitochondria? See, we cannot borrow from the neighbors. The mitochondria cannot cross the cell membrane, so we cannot borrow from the neighbors. The mitochondria has a membrane. It also has the DNA. So we cannot synthesize mitochondria using the existing mitochondria as a template. From where will you get the DNA? So the only answer left is the binary fission. The only answer left is the binary fission. Please understand that mitochondria is basically an endosymbiote. It is a prokaryote which was engulfed by a eukaryote during evolution, and later on they had this symbiosis. Okay, they complement the survival of each other. So the properties of the mitochondria will be similar to that of a prokaryote. And how does a prokaryote multiply? A prokaryote multiplies by binary fission. In other words, you cannot have a de novo synthesis. You cannot have de novo synthesis. All mitochondria comes from pre-existing mitochondria. All mitochondria comes from pre-existing mitochondria. Means if the pre-existing mitochondria has some defect, all the subsequent mitochondria are going to have the same defect. If there is a defect in the mitochondria which is being used for binary fission, then all the subsequent mitochondria will have the same defect. So these two things, all mitochondria comes from pre-existing mitochondria and all mitochondrial maternal origin, gives us the inheritance pattern. These two give us the inheritance pattern. So what is the inheritance pattern? When you look at the inheritance pattern of mitochondrial DNA disorders, we find affected female transmit disease to all offsprings, because the mitochondria is coming from the mother. If the mother has the defective mitochondria, all the offsprings are going to get the defective mitochondria. The affected male cannot transmit disease. The male is not transmitting the mitochondria, so how can it transmit the mitochondrial DNA disorder? So these are the two important points of inheritance pattern. Affected female will transmit disease to all offsprings. Affected male cannot transmit the disease at all. And when you look at the pedigree chart of inheritance pattern, it is very interesting. I'll show you the pedigree chart of inheritance pattern. I'm sure all of you know these symbols. The square or rectangle is the male, the circle is the female, line joining the two represents marriage or consummation, and a shaded symbol throughout, it is shaded, represents an affected individual. So in this case, the female is affected. Now when they are going to have their offsprings, what will happen to the offsprings? One male and one female, what will happen to the offsprings? Remember we have said an affected female transmit disease to all her offsprings. So female is affected, therefore both the offsprings will be affected. In the course of time, these offsprings are going to have their own families. In the course of time, they're going to have their own families. All right, let's say we have a daughter here, a son, another daughter, a son and a daughter. What happens in the first case? The affected male cannot transmit the disease, so the children are unaffected, whereas the affected female will transmit the disease to both her offsprings. So this is the typical inheritance pattern or the pedigree chart that you're going to see in the mitochondrial DNA disorders. The inheritance pattern or the pedigree chart that you're going to see in the mitochondrial DNA disorders. Some examples of mitochondrial DNA disorders. The most common among the mitochondrial DNA disorders is the MELAS. The full name is mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke like symptoms. And this is where we come to one of the very important features, the lactic acidosis. MELAS is the most commonly identified mitochondrial DNA disorder. The full name is mitochondrial myopathy, encephalopathy, and lactic acidosis, stroke-like symptoms. Now, lactic acidosis will be a feature in all the mitochondrial DNA disorders. Please note the lactic acidosis will be the feature in all the mitochondrial DNA disorders. Reason being, the proteins synthesized by the mitochondrial DNA belong to the electron transport chain. So if any of the genes is defective, the electron transport chain will be disrupted. If you block the electron transport chain, what will happen? The aerobic metabolism cannot occur, the pyruvate will accumulate and it will give rise to generation of lactic acid, therefore lactic acidosis. So if we are talking about mitochondrial DNA disorder, number one, you should know the inheritance pattern. Number two, you should know all these patients are going to have the lactic acidosis. Number three, you should know some names. Another example is diabetes mellitus with deafness. Diabetes mellitus with deafness. Then we have Leber's hereditary optic neuropathy. Okay, so these are some of the common disorders and that we see. We have something called Cairn Sayre syndrome. Then we have infantile myopathy. The next class, when we start, I'll give you the complete list. About 11 disorders are there, a total of 11 disorders which are well identified in the mitochondrial disorders. Let's see if we have that list. I can show you that list. Let's see. No, we're not talking about vitamins. We're not talking about vitamins. Let's go back. In the keynote, not the vitamins presentations. Where is the corrected lecture? Okay. Yeah. Diabetes mellitus and deafness, I told you. Leber's hereditary optic neuropathy, the Leigh syndrome, the Leigh syndrome, neuropathy, ataxia, retinitis pigmentosa, and ptosis. No, we cannot call them X-linked recessive disorder. They are not X-linked recessive. They are not X-linked recessive. The inheritance pattern is entirely different from X-linked recessive. They are not X-linked recessive. Myoneurogenic gastrointestinal encephalopathy, myoclonic epilepsy with ragged red fibers, myoclonic epilepsy with ragged red fibers, MELAS, which I told you is the most common. Remember, the mitochondrial inherited disorders are identified very rarely. The mitochondrial inherited disorders are identified very rarely. Among them, the most common, the most common is the MELAS. In addition, we have the chronic progressive external ophthalmoplegia, the Kearns-Sayre syndrome, the Pearson syndrome, and the infantile myopathy. The infantile myopathy. So this is the exhaustive list of the mitochondrial disorders which you must know the names. You should know the names. Details of these disorders are not required at your level. If you know the inheritance pattern, if you know that all of them will have lactase doses, and if you remember the name, that should be enough to answer the question coming from the mitochondrial inherited disorders. So we have completed the DNA part. We have talked about the cell-free DNA. We have talked about the uh, as you can see, majority of the students want to finish today, or actually we already have had almost 4 hour session, so I can understand it is a bit difficult to attend the class via the computer. So Not a problem. We'll continue tomorrow. We'll continue tomorrow, and the timings accordingly will be updated by Dr. Manoj or Dr. Shravan in the morning. So they'll take a call. If you don't have any other class in the daytime, then again maybe